Welcome to this lecture series on eukaryotic gene expression, basics and benefits. This is lecture number 37. Today we are going to talk about a very interesting and very important topic entitled knockout mice. So basically we are going to try to understand how we can knock out the expression of a specific gene in a mammal, whether it is a mouse or rat or embryonic stem cells. The reason why this topic is very important is that if you look at the lectures that we have covered in the last 7 or 8 lectures, we started this part of expression systems series of lectures by looking at how you can express genes in E. coli, and then we discussed about how we can express genes in yeast cells and insect cells. Then we discussed about various eukaryotic protein expression systems, how we can express genes in mammalian cells. Then we talked about human gene therapy, where we were talking about expressing genes in humans with the aim of curing certain genetic disorders. Then we discussed about a very interesting and exciting area of research called DNA vaccines, where a failed gene therapy technique about what is called as a naked DNA injection, which actually failed as a gene therapy technique, found a new application in the form of development of DNA vaccines. Then in the last 2 classes we discussed about generation of transgenic animals and transgenic plants, how we can express genes in mice and other farm animals to make useful recombinant proteins, as well as for understanding certain basic aspects of biology. And we also discussed about transgenic plants, how people are trying to use plants as factories for producing a variety of proteins, recombinant proteins, including what is called as a oral or edible vaccines and so on and so forth. So in all these lectures our emphasis has been primarily on expressing genes, that is how to express an exogenous gene, how to express a transgene in a variety of expression systems. But in today's lecture we are going to cover about how to inhibit or how to repress the expression of a gene in an animal model, and that is what we call as a knockout or knocking out the expression of a gene or knockout mice. So this lecture is different from what we have been discussing in the last 8 lectures. The last 8 lectures the focus has been primarily on expressing a gene in various expression systems, but today we are going to talk about inhibiting the expression of a specific gene. This is not an easy task, because as we know, if you take either a mouse or a human genome, we have more than about 30 to 50,000 protein coding genes, and if you want to specifically inhibit the expression of one gene, that in an animal model, it is a very challenging and daunting task. And in fact the people who developed this technology of knocking out gene in a mouse actually got a Nobel Prize. So let us now try to discuss and see what exactly are these knockout mice. So how knockout mice are generated and how you can specifically knock out the expression of a particular gene in a mouse. So what are knockout mice? A knockout mouse is a mouse in which a specific gene has been inactivated or knocked out by replacing it or disrupting with an artificial piece of DNA. So the loss of gene activity often causes changes in the mouse phenotype and thus provides valuable information on the function of the gene. So the difference between a transgenic mouse model and a knockout mouse model is that in a transgenic mouse we are actually expressing a transgene over the normal genes which are actually present in the animal. We are trying to overexpress a particular gene and see what happens to the phenotype. Whereas in the case of knockout technology, we are actually inhibiting or repressing the expression of a particular gene, so that the gene product is not made, and ask the question, if the gene is not expressed, what happens? So we will discuss as we go about what are the exact differences between transgenic mouse and a knockout mouse. Now the topic we are discussing today is very important, because the researchers who developed this technology to create knockout mice actually won the Nobel Prize in the year 2007. So the Nobel Prize for Physiology and Medicine in 2007 was actually awarded jointly to Mario Capicci, Martin J. Evans and Oliver Smithies for their discoveries of principles for introducing specific gene modifications in mice by the use of embryonic stem cells. Why were they given this Nobel Prize for this discovery? Because the ability to delete or mutate any gene of interest in mice has transformed the landscape of mammalian biology research. So what I am going to discuss in the next few minutes is to give you evidence that how this technology developed by these 3 people about the ability to knock out genes in embryonic stem cells and then take these embryonic stem cells in which the genes have been knocked out and then put them in a mouse embryo and create a mouse, what is called as a knockout mouse, in which that particular gene is not expressed. This creation of this knockout mouse technology has changed the way the biological systems function, the way gene functions can be studied, and so on and so forth. And that is why this knockout mice played a very important, considered as a very important discovery in biology, and these people got the Nobel Prize. So what exactly these people did? Martin Evans actually spent a lot of time and published a number of papers and standardized the technology for the cultivation of embryonic stem cells. And Oliver Smithies was the first to use these embryonic stem cells and developed techniques for targeting a transgene to a specific region in the ES cells. He could demonstrate that it is possible to target a transgene to a specific locus. As we have discussed in the earlier lecture, especially in human gene therapy, the problem with things like retroviral vectors is that the retrovirus goes and randomly integrates anywhere in the genome and causes a lot of problems, and in fact it even caused cancer in a couple of instances. So it is very difficult to specifically target a gene to a specific region on the locus in the mammalian genome, and because whenever you put a transgene, the chances of random integration are much higher than the targeting to a specific region by homologous recombination. So Oliver Smithies and Mario Capici actually developed technologies to selectively identify those cells in which gene targeting has taken place compared to those in which the gene has gone and randomly integrated elsewhere in the genome. So some of the key papers published by Oliver Smithies was which are just mentioned here, where for example in 1987 he actually published a paper on targeted correction of a mutant HPRT gene in mouse ES cells, and he stated that this modification of a chosen gene in pluripotent embryonic stem cells demonstrates the feasibility of this route to manipulate mammalian genomes in a predetermined way. And in 1985 he published another papers where he actually showed insertion of DNA sequence into human chromosomal beta globin locus by homologous recombination, and then germline transmission of a planned alteration made in HPRT gene by homologous recombination in embryonic stem cells. We will discuss some of these papers in detail as we go along. Similarly, Capici published many landmark papers where in 1987, around the same time as Oliver Smithies published, he showed actually site-directed mutagenesis can be made by gene targeting in mouse derived embryonic stem cells, and 3 years later he actually demonstrated using this technology it is actually possible to disrupt the function of a proto-oncogene called INT1, leading to severe abnormalities in midbrain and cerebellar development. INT gene is actually a proto-oncogene, plays a very important role in development, and if you knock out the expression of this INT1 gene, it results in several developmental abnormalities. So these are some of the key papers published by these Nobel laureates in the area of knockout mice. Now what is the importance of knockout mice in biology and medicine, and why this technology became so popular, and why these individuals got Nobel Prize? Now knocking out the activity of a gene provides valuable information about the functions of the gene. Since many genes are common between humans and mice, knockout mice gives the researchers information about the function of the gene in humans. So far the difference between a transgenic mouse and a knockout mouse is that in the case of a transgenic mouse, we are overexpressing a gene over and above that of a normal or mutant copy which is present, whereas in the case of a knockout mice, we are blocking the expression of that particular gene and asking the question, what happens if the gene is not expressed? Does it have an important role? What happens, and so on and so forth. So knockout mice has been very useful in studying and modeling different kinds of cancer, obesity, heart disease, diabetes, arthritis, a number of diseases, and knockout mice also offer a biological context in which drugs and other therapies can be developed and tested. So I am going to give a few examples as we go along to see how the development of this technology for knocking out genes in mice revolutionized research in biology and medicine. So let us now spend some time to understand what is the main difference between a transgenic mouse and a knockout mouse. Now a transgenic mouse typically expresses one or more copies of a gene that is integrated into genome in a random fashion. This is what we have shown, because when you inject a transgene into the fertilized egg of a mouse, it goes and randomly integrates in a head to tail fashion in anywhere in the mouse genome, so we have no control over it. Whereas in the case of a knockout mouse, both alleles of a gene are deleted in a targeted fashion by homologous recombination. So in the knockout mouse, since you have to specifically knock out a specific gene, you have to target your transgene construct such a way that it goes and recombines with the gene present in the chromosome of the cells, so that genetic recombination takes place and this goes and specifically knocks down the expression of that particular transgene. So while traditional transgenic mice are generated to express a protein, much information can be learned from the elimination of a gene or deletion of a functional domain of a protein. So, this is what the main distinction between a transgenic mouse and knockout mouse. In a transgenic mouse model, you are trying to understand the function of a gene by actually overexpressing the gene, by overexpressing the protein coded by the gene, and ask the question what happens when you overexpress that particular gene or protein. For example, we had learnt in the last class that if you overexpress the human growth hormone or the rat growth hormone gene in the mouse, it grows much bigger, because growth hormone promotes growth using a methylothionein promoter. So, the creation of a transgenic mice involves injection of DNA into fertilized eggs and looking for adult animals that produce their protein of interest. That is what we discussed in the previous class. Whereas in the case of transgenic mice, the transgene gets inserted into the genome randomly. These are the two important features of a transgenic mouse. So, transgenic mice are actually used primarily to gain of function mutations, wherein two copies of the normal gene are joined by additional copies which would overproduce the normal or mutant protein. So, this is what we understood by the transgenic mice. So, the normal copies or a mutated copy are still there, but over and above this normal copies are present in their normal location in the genome, we are adding extra copies by overexpressing the gene and generating transgenic mice, and ask the question what happens when you overexpress the gene, what is the phenotype you generate. So, it is a gain of function mutation that we are studying. So, using transgenic mice, one can infer a gene's function by observing what happens when there was too much of a protein or when a mutant copy interfered with the normal one. So, this is basically what knowledge we gain by making transgenic mice. Whereas, in the case of knockout mice, the knockout technology allowed research to delete specific genes. So, in this case, the transgene, which is usually a mutant or deleted version of the normal gene, is introduced into the embryonic mouse cells in culture and then allowed to recombine with the sister DNA in the genome. So, you make what is called as a knockout construct, in which the gene is either disrupted or mutated, and when you introduce this mutated version or a deleted version of a gene inside your cells, by using specific techniques, you select for those cells in which this transgene has recombined with the endogenous chromosomal copy and therefore has deleted both the normal alleles in the chromosome, so that only your transgene or the mutant version of the gene is expressed. So, you do not have the normal copies of the gene anymore. It is the primary distinction between a transgenic mouse and a normal mouse, the knockout mouse. So, the recombination cuts out the resident DNA and inserts the mutant DNA, which can then form the damaged protein or does not produce any protein at all, depending upon how you have designed your transgene construct. The embryonic stem cells then grow into a whole mouse with a specific mutation exactly where it is supposed to be in the genome. So, this is the major difference between transgenic mouse and knockout mouse, is that in the case of a knockout mouse, your construct has gone exactly the same place where the normal counterpart is present. Whereas, in the case of a transgenic mouse, your gene may go and randomly integrate anywhere, and the normal copy may be still there elsewhere in the normal place of the genome, but your transgene may be getting expressed somewhere else from the genome. So, the gene knockout technology allowed researchers to study loss of function mutations, wherein one can infer gene's function by observing what happens when the gene is absent or when mutant copy of the gene is expressed instead of the normal one. So, by generating transgenic mice, you understand the gene function by gain of function mutations, whereas by generating a knockout mice for the same gene, you are asking the question what happens when the gene is not expressed, when the function of the gene is lost. So, it is a loss of function mutation. So, transgenic mice is a gain of function mutation, knockout mice is a loss of function mutation. And the first human disease models created with this technology was for cystic fibrosis from the labs of Smithies and Evans. We will discuss some of these things. So, I am going to give you a series of slides just to emphasize the point that what kind of experiments were actually done, what kinds of genes have been knocked out. There are a number of genes have been knocked out. I am going to give only a selected list of examples. I am going to show some very nice pictures of how knocking out specific genes, what kind of phenotype people got, and how you can demonstrate that this gene has a particular function by using the knockout technology. Here is, for example, one picture where it shows a laboratory mouse in which a gene affecting hair growth has been knocked out. The one on the left, so it has much less hair compared to the wild type mouse. So, clearly saying that this gene is very important for the hair growth. If you knock out the gene, you get a mouse which does not have as much hair as the normal one. Here is another example. Here is another gene. If you knock out this gene in this mouse, the mouse becomes obese. You can see this is a normal mouse, and you can see this mouse is much bigger. So, this is a knockout mouse model for obesity, and you knock out that particular gene, the mouse becomes obese or fat compared to the normal, clearly telling that this gene has a very important role in obesity. It is another very interesting gene. a gene called NHLH2, which is actually a transcription factor, and if you knock out this expression, it results in what is called say adult onset obesity. Now what is this? You can see when the mice, this wild type mouse and a knockout mouse, when they are 8-week old, they both look normal, but as they age 12 weeks, 28 weeks, 52 weeks, as you can see, the mouse in which this particular gene has been deleted is becoming fatter and fatter. It is becoming more obese compared to the normal mouse. So as the mouse age, they become more and more obese. So this is called as a adult onset obesity, indicating that this transcription factor somehow is playing a very important role in this particular disease. So it gives an idea, understand what kind of genes are getting affected by knocking out the expression of this particular gene. So NHLH2 knockout mice at 8 weeks of age are similar to the normal mouse in weight and body structure, but older knockout animals have an increase in abdominal fat and body weight. So very interesting phenotypes you can observe by knocking out these very important genes. Here is another example. If you knock out a gene coding for a protein called GDF8 known as myostatin, and this mouse has twice the muscle mass compared to the wild type. So the muscle mass increases. Very interesting, clearly saying that this protein plays a very important role in myogenesis or muscle formation. Another important mouse, FGF5, if you knock out this particular protein, the mouse has long angora like hair. So very interesting phenotypes. Again, if you knock out a gene that codes for a DNA methyltransferase gene, it results in embryonic lethality. Again published by Roloff Janisch groups in Cell in 1992. As we studied, we had studied how epigenetics plays a very important role in embryonic development. We had discussed extensively how DNA methylation plays a very important role, especially de novo methylation takes place in the fertilized egg, and then a new methylation pattern is established as the fertilized egg starts dividing during early embryonic development stage, and there are what are called as DNA methyltransferase gene, which actually is plays a very important role in methylation of the promoters and turning on what are called as turning on or turning off specific genes during embryonic development. And to actually demonstrate that this DNA methyltransferase or DNA methylation plays a very important role in embryonic development, they actually knocked out the DNA methyltransferase gene, and although the embryonic cells did not show any phenotype, they were normal. That is, if you knock out the DNA methyltransferase gene in embryonic stem cells, they still divide and behave normal, but when when you take these AES cells in which the DNA methyltransferase gene is knocked out and you develop a mouse model, the mouse did not develop beyond a certain stage of development, clearly indicating that DNA methylation, if you knock out, if DNA methylation is not proper, embryonic development is totally abolished. You cannot get an adult mouse out of it, indicating DNA methylation plays a very important role during embryonic development. Again, P53 gene. So P53 is a tumor suppressor gene, and we all know people have done number of experiments in number of cell lines and then show if you knock out P53 gene, the cell line becomes cancerous, it becomes tumorigenic. Okay, but when they actually knocked out the P53 gene in mouse, the P53 knockout mouse developed normally, but developed a variety of cancers including lymphomas as they grew old. So by actually demonstrating in an animal model that if you knock out P53, it ultimately develops cancer, it provided ultimate proof that yes, P53 is actually a tumor suppressor gene and is essential for the normal phenotype of an individual. If you mutate or if you delete the P53, it leads to cancer. One important result of the gene knockout models revealed is that cells need more than one mutant gene to become cancerous. When you knock out P53, it actually took many months for cancer to arise. So thus cells need more genetic alterations than just one. So although ultimately the mouse developed cancer, but in the initial stages of development the mice were pretty normal, all the P53 was knocked out, but as they age, as they grew older, more and more mutations accumulated, and therefore along with mutant P53 or deletion of P53, other mutations are also required for getting a cancer phenotype. Similarly, another very important tumor suppressor gene called the retinoblastoma gene, when they actually knocked it out, people actually expected that these mice should form tumors in the eye, because that is what retinoblastoma is. But instead of eye tumors, these animals actually got pituitary and thyroid gland cancers, which was totally unexpected results. So this knockout technology actually gave phenotypes which were not expected. Many of these knockout mice developed phenotypes which were very interesting and which totally were contradictory to what was actually expected of these knocking out of these genes. and it was later found that a second gene actually protected the eye cells from cancer, and both mutations were required to form tumors. So the knockout models, animal models, gave tremendous knowledge on the function of these genes, and in many cases either substantiated or provided the ultimate evidence of all the work that was done in cell lines, and many times it actually gave up some new very interesting and surprising results, making you to develop new lines of research. Similarly, in another study, when the gene encoding telomerase is a protein that is responsible for maintaining the tips of the chromosomes, and as long as telomerase is there, the chromosomes replicate normally and the tips replicate, but if you knock out this telomerase, then the ends of the chromosomes cannot replicate anymore, and in such mice cancers arose when chromosome tips shorten over a course of lifetime of cell division, and the failing tips cause chromosomes to break and recombine incorrectly until finally the cells reach a large number of mutations needed for transition to malignancy. But what all these knockouts in these genes involved in cancer actually told is that the most important factor that drives cancer is age. So whenever they mutated many of these oncogenes or tumor suppressor genes, it is not that the animals developed cancer immediately. It is only over a period of time, as the mice age, they started developing cancer, indicating that accumulation of mutations, you require more than one mutation for cancer to arise. So the knockout mice models also contributed to the understanding of the function of genes in development. In fact, very often oncogenes were found to be critical for normal development. As I told you in the one of the first slides, Mario Capicci actually, when he knocked out the INT2 gene, which is actually a proto-oncogene, it led to a lot of development abnormalities. So it became very clear that many of these proto-oncogenes, which actually are involved in cancer development, they play a very important role during embryonic development. So these kind of a very interesting results came out of knockout mice research. So I have so far discussed with you what is the importance of knockout technology and how the knockout technology is different from that of a transgenic technology. In the case of transgenic animals, you are actually overexpressing the gene over the normal gene which is present in the chromosomal copy, and the gene that you are introducing actually can randomly integrate anywhere in the genome and express it. Whereas in the case of a knockout technology, the transgene that you are introducing goes specifically to a particular locus in the genome, recombines with the normal copy present in the chromosome and replaces the normal gene present in the chromosome. So your transgene is present exactly in the same place as the normal gene is present. So it is very specific. So site specific recombination, the transgene goes and replaces the endogenous gene by site specific recombination, and so it is very specific. The transgenic mice, you are looking at the function of a gene by gain of function mutations, whereas in the case of a knockout mice, you are trying to understand the function of a gene by loss of function mutations. These are the two major distinctions between transgenic mice and knockout mice, and I have given you several examples as to how generating knockout mice or by knocking out specific genes, how very important, very interesting phenotypes could be developed, whether you knock out a transcription factor or whether you knock out a proto-oncogene. You can see mice develop very interesting phenotypes, and by knocking out these kinds of genes, one can also understand the biology of the disease much better, and you can also develop animal models for a number of diseases. So now let us spend some time to understand what is the technology. How can you knock out a specific gene? It is almost like needle in a haystack. There are so many genes in the genome, but you want to specifically knock out a particular gene. How do you do that? Because genetic recombination is a very rare event, and the transgene that you have introduced has to go and specifically recombine with the endogenous gene which is present in the chromosomal copy. It happens only 1 in 1000 or 1 in 10,000. Usually it goes and randomly integrates anywhere in the genome. So the challenge in developing a knockout mouse is to identify those rare recombination events rather than over the background of random integration events. So you need to kill those cells in which your transgene or your knockout construct has got randomly integrated all over the genome, and select only those cells in which your knockout construct or transgene has specifically gone to replace the endogenous gene by genetic recombination. So you have to score for this rare recombination event over the large background of random integration events. That is the key feature of this knockout technology. So if you want to generate a knockout mouse, you need to, the gene that you have introduced has to undergo what is called as a homologous recombination. So what are the various steps in generating a knockout mouse? The first is you need to design what is called as a knockout construct. Just as if you want to express a gene and generate a transgenic construct, there you need a promoter, and then you put your gene of interest downstream of the promoter and put the proper polylinear sequences, and then you inject them into fertilized egg. It goes and randomly integrates, and the promoter will now drive the expression of your transgene, and your protein is overproduced. Whereas in the case of the knockout construct, the gene that you have introduced actually should be a mutated version, or certain regions of the gene should be deleted so that it is a non-functional gene. So the knockout construct is different from a transgenic construct. In the transgenic construct, you are actually the normal version of the gene is usually expressed to make the wild type protein, whereas in the case of a knockout construct, usually the gene that you are introducing will either carry a mutation or it will actually carry a deletion of certain region. And once you make a knockout construct, you introduce the knockout construct into a mouse embryonic stem cells in culture, and then screen the embryonic stem cells and select only those whose DNA includes the new gene, and then implant the selected cells into normal mouse embryos, making a chimeric mouse, and then you breed these chimeric mouse to generate homozygous. Implant the chimeric embryos into pseudopregnant females, and then once the females give rise to birth to generate chimeric mice, you then breed them to generate a pure line or a homozygous knockout mouse strain. These are the various steps in generating a knockout mouse, but let us now go step by step and try to understand how exactly go about developing a knockout mouse. So the key mechanism involved in generating a knockout mouse is homologous recombination. I am sure all of you have studied what is homologous recombination. Homologous recombination normally takes place when the maternal and paternal chromosome come together, especially during meiosis, and exchange parts of their chromosome. This is what is the molecular basis for the differentiation, which usually happens during the meiosis in the germ cells. So you can see that the paternal and maternal chromosome come together, what is called as a genetic recombination takes place, that the very famous Holiday junction is formed, and segments of DNA gets exchanged between the two chromosomes, and you get a recombinant chromosome. But this homologous recombination is a very rare event. So what happens during the development of a knockout? The gene knockout is actually accomplished by homologous recombination involving a double crossover event. That means you need to have crossovers at two different regions of the genes. Now I am going to explain in a schematic manner, in a very simple manner, how exactly the gene knockout takes place. Now let us see, this is the gene of your interest, let us say, and you want to actually introduce a mutation in this particular region of the gene. Now so you have to now design a knockout construct where you either PCR amplify or isolate the gene of your interest, and then in the very crucial region of this gene, which is very important for the function of the gene, you replace that portion of a gene with an expression cassette that codes for a resistance for a particular drug. It could be neomycin or usually an antibiotic resistance gene. So you replace the essential region of your gene of interest with an expression cassette that codes for a antibiotic resistance marker. So once you have, this is what is called as a knockout construct. Now once you have a knockout construct, you then introduce this knockout construct into embryonic stem cells. So in the embryonic stem cells, inside the nucleus, you have the normal copy, let us say which is homologous to this gene. Now homologous recombination takes place now between your transgene, the gene you have introduced knockout construct, and the endogenous gene. So this crucial region of this gene will now get replaced by the drug marker, and therefore you now get a disrupted gene. So this normal gene now got disrupted because the region which is very crucial for the gene function has now been replaced by an expression cassette coding for a drug resistance marker, and then you can now select those cells which have undergone recombination by screening for drug resistance. This is a very general strategy involved in generating a knockout embryonic stem cells. Now you take this embryonic stem cells, screen for those in which the recombination takes place, and then put them in pseudopregnant females, and you get a knockout mouse. I will give a specific example. For example, if you want to now knock out the hypoxanthine guanine phosphoribosyl transferase HPRT, which is very important in the salvage pathway of purine metabolism, what you design is that you design a knockout construct wherein you introduce a neomycin resistant expression cassette between 7th exon and 9th exon, so that the 8th exon is missing. So this is your transgene construct. Now what happens when you now take this transgene construct and introduce into embryonic stem cells? Recombination takes place between your transgene and the endogenous chromosomal copy of the gene. Due to the double crossover, you now get a knockout of the endogenous chromosomal copy gets knocked out. So, in the place of the 8th exon, now you got a neomycin resistant Cas, and you can now see this now becomes HPRT minus and G418 resistant. That means these cells now are expressing neomycin resistance gene marker, therefore they become resistant to G418, but since the 8th exon is missing, they cannot make the functional HGPRT protein, therefore they become HPRT minus. So, this is the basic strategy for knocking out a particular gene. So, the certain crucial regions of the exons are replaced by a selection marker, and then you screen for the particular cells in which the selection marker is expressed, and those cells are the ones in which the gene has been knocked out. As I said, the crucial step in generating a knockout mouse is to distinguish a random integration event from a cells those undergone homologous recombination. As I said, homologous recombination is a very rare event. Usually whenever you introduce a transgene into the cells, it goes randomly integrates all over the genome. So, the key to developing a knockout cells, ES cells or knockout mouse is to score for those cells which have undergone homologous recombination and kill those cells in which your transgene has got randomly integrated elsewhere in the genome. This is carried out by doing what is called as a using positive and negative selection markers. It's a very important feature. So, I am going to spend some time to explain to you how do you do this positive and negative selection, and how you can selectively select those cells in which have undergone genetic recombination, whereas and kill those cells in which the gene has got integrated randomly. So, how do we distinguish between random integration and homologous recombination? This is carried out by doing what is called as positive and negative selection markers. Now, what is this? Positive selection markers are those which are used to enrich recombination events. The expression cassettes encoding antibiotic resistance are usually employed as positive selection markers. So, suppose you have a gene which has say number of exons, and suppose you want, and let us say exon 2 is crucial for the function of that particular gene. What you do is that you design a knockout construct such a way that the exon 2 is replaced by a positive selection cassette, and in this case it is a neomycin expression cassette. So, the exon 2 of this gene has been replaced with a neomycin resistance cassette. So, you have on either side of the neomycin resistant cassette you have exon 1 and exon 3. So, these are the regions of homology, and these are the regions which are crucial for recombination, the double crossover that has to take place between the endogenous chromosomal copy when you introduce this construct into the cells. Now, what is negative selection markers? Negative selection markers are used to enrich for homologous recombination events over random insertions. Usually, the herpes simplex virus thymidine kinase coupled to ganciclovir treatment is actually used as a negative selection marker. Now, what is this? Ganciclovir is actually a synthetic nucleoside analog of deoxyguanosine, which is phosphorylated by this viral thymidine kinase, and once it is phosphorylated, and when this phosphorylated form of nucleoside analog gets incorporated into the elongating DNA chain during DNA replication, it inhibits DNA by chain termination, and therefore DNA replication stops. So, ganciclovir is a nucleotide analog which when incorporated in elongated DNA synthesis cannot proceed further, and therefore the cells die, cells cannot divide anymore. So, HSV-TK gene preferentially incorporates ganciclovir into the DNA, and the host thymidine kinase or the mammalian thymidine kinase has no affinity for ganciclovir. So, only those cells which express the viral thymidine kinase, ganciclovir will get incorporated into DNA, and those cells die because they cannot divide. And these kind of a negative selection markers are usually incorporated into a knockout construct outside the region of homology. Very important, the positive selection marker is incorporated into the knockout construct within the regions of homology, whereas the negative selection cassette is incorporated into the transgene in the knockout construct outside the region of the homology. So, this is basically the knockout construct that you have to make if you want to knock out a particular gene. So, what happens once you make this knockout construct, you inject this knockout construct to embryonic stem cells, recombination now takes place between these two regions of homology, and as a result only this region now gets replaced. So, you will now the thymidine kinase gene will not get incorporated into the chromosomal locus, only the neomycin resistant cassette will get incorporated if the cells have undergone homologous recombination. So, such cells will be neomycin resistant, but since they do not carry the HSV thymidine kinase gene, they are resistant for G418 or ganciclovir resistant. So, if you add ganciclovir, these cells will not get killed. On the other hand, if random integration has taken place, if there is no homologous recombination here, if the entire transgene has gotten inside, got integrated by randomly, then both the neomycin gene as well as the thymidine kinase will get incorporated, and as a result, these cells in which this entire cassette has got integrated because of random integration will be both neomycin resistant as well as HSV-TK positive. They will also be expressing the thymidine kinase. Therefore, they will be resistant to G418 or ganciclovir, G418, but they are sensitive to ganciclovir. So, if you now take those cells in which have undergone random integration, that means in these cells both neomycin resistant cassette as well as thymidine kinase will express both the neomycin resistance marker as well as the thymidine kinase, and therefore such cells, if you now treat with G418 and ganciclovir, ganciclovir now gets incorporated into the DNA and these cells will get killed. This is what is called as a negative selection. Whereas those cells in which have undergone homologous recombination, thymidine kinase gene will not get incorporated, only the neomycin cassette will get inside. Therefore, there will be G418 resistant, but also resistant to ganciclovir because they do not express HSV-TK, and therefore ganciclovir will not get incorporated into the DNA. Therefore, the cells cannot be killed with ganciclovir. So, you can see by making this kind of a knockout construct, you can now select only those cells which have undergone in homologous recombination, because these cells will be resistant to both G418 as well as ganciclovir. Whereas in those cells where the transgene has got randomly integrated everywhere, they will be expressing both neomycin resistant marker as well as HSV-TK, and therefore they will be G418 resistant because of the neomycin resistant. They will also be sensitive to ganciclovir because they are expressing thymidine kinase. And when you treat them with these two compounds, all those cells in which the random integration taking place, they all die. So, you can see by using this very clever strategy, you can select only those embryonic stem cells which have undergone targeted disruption of your gene of interest, whereas all those cells in which the gene has got randomly integrated, they get killed. So, this is called as a positive and negative selection marker, and using this kind of a technology, you can identify only those ES cells which have undergone genetic recombination or which have undergone targeted disruption. And you can actually once you have those ES cells, you can actually confirm that target disruption actually taking place by actually designing primers and doing a PCR. Usually you design one primer outside the region of homology, one primer within your selection marker, and by doing a PCR, and if you generate the expected size of fragment, you know that yes, your transgene has really gone to the right place. You can also confirm by doing what is called southern blot analysis. For example, I have given an example here. Let us say there is an EcoR1 site, EcoR5 site flanking the positive selection marker. Then suppose the antibiotic resistant gene also has an EcoR5 site. Then if you now isolate the genomic DNA from these ES cells and then do a southern blot analysis using a probe somewhere in this region, this probe will now hybridize to a 4 kb region as shown here. Whereas if the disruption has not taken place, it will generate the entire end, because this EcoR5 site is not there. Therefore, 4 plus 5, there are only 2 EcoR1 sites. This probe will now hybridize to a 9 kb region, where that means these are not the knockout, the recombination has not taken place, whereas if recombination has taken place, because you are introducing an extra EcoR5, this probe will hybridize only to a 4 kb band. Therefore, all those which shows a 4 kb band in addition to a 10 kb band, remember you are only knocking out one allele. So, these are heterozygous. So, in addition to the normal copy of 10 kb, you will also generate a additional 4 kb, indicating that one of the alleles has undergone homologous recombination, and therefore, you can confirm target disruption. So, then you take the CS cells, now introduce into the mice, and you generate a chimeric mice, and then you chimeric mice, if you now breed, then you get a homozygous knockout, and in such knockouts, if you now do a southern blot analysis, you will not get the 9 kb, you will see only the 4 kb, indicating that both alleles has now been knocked out. So, this is the summary of the gene targeting strategy, which was developed by Mario Capicchi, the positive and negative selection protocol. So, the targeting construct usually contains a neomycin resistant cassette in a crucial exon that is required for that particular gene, and If the cells are neomycin resistant and HSV-TK minus, that means those cells have undergone gene targeting, the gene has been disrupted, and such cells will be resistant to G418 because they are expressing neomycin, but they will also be resistant for ganciclovir or FIAU, which is an analog of same, both are more or less same, because they are not expressing the HSV thymidine kinase gene. Whereas in those cells where the transgene has got randomly integrated, both neomycin resistant marker and HSV-TK marker will get integrated in the chromosomal copy, and therefore they will express both neomycin resistance as well as HSV-TK, and such cells will be G418 resistant but sensitive to ganciclovir or FIAU, so they get killed. So by using this positive and negative selection, you can score for only those ES cells which have undergone targeted disruption of your gene, whereas all those cells in which we have undergone random integration, they are eliminated. So this is basically the technology. Now what are the major drawbacks of this knockout mouse? One of the major drawbacks of generating knockout mouse is that many genes that participate in interesting genetic pathways are essential for either mouse development, viability or fertility. They are all very crucial. Therefore, a traditional knockout of a gene can never lead to the establishment of a knockout mouse strain for analysis. For example, I want to understand how important is a homeotic gene. Now, as you know, homeotic genes are very important for normal development. We have studied it in detail in some of the previous classes. But if I now knock out a homeotic gene, since without that homeotic gene the embryonic development will not proceed, I will never get a normal embryo. So it is very difficult for me to establish a mouse line, a knockout mouse line, because those mice will never develop beyond a certain embryonic stage, because the homeotic gene is crucial for it. So one of the major drawbacks of this knockout mice is that genes which are crucial for embryonic development, in such cases you cannot develop these knockout mouse cell lines because they are all embryonic lethal, and such embryos die very early during their development. So about 15% of the gene knockouts are developmentally lethal and therefore cannot grow into adult mice. Therefore it becomes difficult to determine the function of these genes in adults. So see, many of these genes are not only important in development stage, they may have a different function during the adult life. So you can never understand the complete function of these genes because they are all embryonic lethal. So to overcome this major drawback of this knockout mouse, you develop what is called as a conditional knockout approach, which allowed researchers to delete the gene of interest in a time and space dependent manner using what are called as site specific recombinases. So instead of knocking out the gene in all the cells of your body, you use what are called as site specific recombinases, so that your gene gets knocked out only in select tissues, or your gene gets knocked out only when you want them to be knocked out. For example, I want to knock out the function of a gene at specific stage during development, it is possible. This is what is called as a conditional knockout. Let us now see how is this conditional knockout is done. So using the site specific recombinases, it is possible to knock out the expression of a gene in a specific mouse tissue or at a specific stage of development or in response to a specific inducer. So I am going to spend some time to explain to you how this conditional knockout is done, because it's a very important technology. Now the key to this development of this conditional knockout mouse technology is the identification of site specific recombinases. One such site specific recombinase was identified very early during the 1980s is by Brian Sawyer, who identified what is called a Cre-Loxp system, for which is actually a recombinase which is present in a bacteriophage P1. Okay. What actually they showed is that by using this Cre-Loxp system, you can actually knock out genes conditionally in transgenic mice. This was shown by Rajewski's lab in 1995. The original papers are actually shown here. So let us now see what is this Cre-Loxp system, how exactly you can knock down genes specifically in select tissues or at specific period of development using this site specific recombinase. Now Cre recombinase is a site specific integrase isolated from the P1 bacteriophage. It catalyzes recombination between two of its consensus DNA recognition sites known as the Loxp sites, which are 34 base pairs in length. Here is the Loxp site I have shown here. So the Loxp site, that is the site that is recognized by this Cre recombinase, consists of two 13 base pair palindromic sequences I have shown here. These are palindromes, you can see ATAACTT, ATAACTT. So they are in a inverted fashion, just like the steroid hormone response elements which you have studied in earlier classes. So the recognition sequence for this Cre recombinase known as the Loxp site consists of an inverted repeat or a palindromic sequence separated by an 8 base pair sequence. Remember, the steroid hormone response elements, for example, if you take the glucocorticoid response element which you have studied, also consists of an inverted repeat AGA, ACA, TGT, CCT separated by 3 base pair spacer, so that 2 monomers of the glucocorticoid receptor can bind to these sites and then activate transcription. The same way, the recognition sequence for the Cre recombinase also consists of 2 inverted repeats separated by a 8 base pair spacer. So, what happens when your Cre recombinase binds to this sequence? Now, when these 2 LOXP sites are placed in a trans orientation, that is when they are present as direct repeats, the functional recombination removes the functionality and knocks out whatever is there in between. So, when you have 2 LOXP sites in the same orientation, when Cre recombinase is expressed in such cells, now the Cre recombinase promotes homologous recombination and whatever region that is present between these 2 sites is removed, and therefore, that region is lost. So, if you introduce 2 LOXP sites in this orientation in a gene, this region of that particular gene will be deleted and therefore, you will get a non-functional gene. On the contrary, if the LOXP sites are present in a cis orientation, that is they are in the opposite orientation, now when the Cre recombinase is expressed in such cells, it now inverts the gene sequence. The gene which is present in 5' to 3 direction now gets inverted in the opposite direction. On the other hand, if you place the 2 LOXP sites in 2 different chromosomes, then it can bring about chromosomal translocation, and as you can see, so depending upon how the LOXP sites are arranged, whether they are arranged in a cis orientation, trans orientation or on 2 different chromosomes, you can get different types of genotypes. In one case, the region between the 2 LOXP sites can be deleted, or the region between the 2 LOXP can be inverted, or chromosomal translocation can be brought about by the Cre recombinase depending upon how these LOXP sites are arranged in a particular genome. So, this is a cartoon where I have actually tell you what exactly happens. So, if you want to know knockout using a Cre recombinase, what you do, you introduce the 2 LOXP sites along with a neomycin resistance marker, usually in the intronic region of a gene of your interest. Now, recombination now takes place between the mouse genome, and this is the mouse genome containing the normal gene. This is your knockout construct where you have the 2 LOXP sites and a neomycin resistance marker, and now you get embryonic stem cells in which the neomycin expression cassette is incorporated and you have the LOXP sites. Remember, the gene is still normal because all the exons are still there, but since they are expressing neomycin resistant, you can actually select for these cells. Now, take these ES cells, now introduce into the pseudo pregnant females and you generate a chimeric mouse, then you breed this chimeric mouse and you get what is called say homozygous mouse in which both the alleles now contains the LOXP sites. These are called as knockout ready mice. Remember, the gene of your interest is still normal because it still has all the exons. In addition, it is also expressing the neomycin because the neomycin resistant cassette is in the intronic region. Now, if you now cross this homozygous knockout ready mouse with a mouse that is actually expressing Cre recombinase, then Cre recombinase now will remove the LOXP sites between the 2, and now the exonic region is now removed, the 2 and 3 are removed now. So, now you have a non-functional gene. So, now we can disrupt a gene. So, the mice are normal as long as Cre recombinase is not expressed, but once you breed this mouse with another mouse which is expressing Cre recombinase, now Cre recombinase now promotes recombination and removes the region between the 2 LOXP sites, and you now get a conditional knockout mouse. This is conditional because the key here is that you can now express the Cre recombinase in a specific manner. You can put the Cre recombinase under a tissue specific promoter or a promoter under an inducible promoter or a developmental space specific promoter. So, depending on how you express Cre recombinase, you can target disruption can be brought about only in those situations. For example, you express the Cre recombinase only in select liver tissue, then only in liver tissue your gene will be disrupted. Or if you express the Cre recombinase in a inducer specific promoter, in an inducible promoter, only when you add the inducer, your gene will be disrupted. So, this is what is called as a conditional knockout. So, spatial and temporal knockout is achieved by the choice of the promoter used to drive the Cre gene expression. Several mouse lines expressing Cre from a promoter that is either tissue specific, cell specific, developmentally specific or responsive to an exogenous like tetracycline are now available, and thus several promoter specific mouse models can be generated. So, these are all available. For example, there is a consortium which actually generated all these, there are now mice, conditional knockout mouse models where the Cre recombinase is expressed under a specific tissue specific promoter, and therefore, only in those genes when you now cross these mice with the mice containing the LOX sites, only in those tissues in which Cre recombinase is expressed the knockout takes place. So, this is the difference between a conditional knockout and a regular knockout. In the regular knockout mice, in all the cells of the body the gene is knocked out, whereas in the conditional knockout only those tissues in which Cre recombinase is expressed the gene will be knocked out. So, depending upon whether you are using a cell specific tissue specific promoter or a developmental specific promoter or an inducible promoter, you can bring about knockout only in a conditional manner. For example, instead of expressing a Cre recombinase in a tissue specific manner, you can also take the Cre gene, recombinase gene, and fuse it to the ligand binding domain of estrogen receptor. So, when you now develop those mice in which the Cre recombinase is bound to the estrogen receptor, now this Cre recombinase will be present in the cytoplasm when there is no hormone. The moment you inject hormone to such mice, now the Cre recombinase goes inside the nucleus and brings about recombination with the two LOX sites and you get targeted repression. So, estrogen inducible knockout can be brought about. So, in these mice your target gene of interest will be knocked out in an estrogen or a tamoxifen dependent manner. So, either by using tissue specific or developmental specific reservoir or using this kind of inducible system, you can conditionally knock out your gene of interest in select tissues or whenever you want. So, this is what is called as a conditionable knockout. There are actually consortia now. For example, here is a European conditional mouse mutagenesis program where they have developed several such mouse lines which are expressing Cre under a wide variety of tissue specific promoters. For example, there are mice which express Cre only in the central nervous system. There are mice which express only in the liver using the alpha antitrypsin or albumin promoter. There are mice in which the Cre recombinase is expressed only in adipocytes promoter or only in the intestinal epithelium or only prostate, only male germ cells, skeletal muscle, keratinocytes. So, using these kind of specific tissue specific promoters, you can regulate the expression of the Cre recombinase, and when you take such mice and cross with another mice in which you have your gene of interest in the two LOX sites, your gene will be disrupted only in those tissues in which Cre recombinase is expressed. So, this is how conditional knockout can be brought about. In addition to Cre recombinase, there are also several other recombinases. Here I am not going to go into details. There are other recombinases, for example, Flic recombinase, which recognizes what are called F40 sites. There are also many other recombinases like DRE, PHXC31 and so on and so forth. The mechanism is more or less similar to what I have expressed for Cre recombinase. So, you can see this development of this conditional knockout has revolutionized the way we can study genes and understand their functions in animal models. I just want to introduce one more terminology. So far we have been talking about knockout. There is also what is called as a knock-in. Now, in contrast to knockout in which the gene or a part of a gene is deleted, in the case of a knock-in, the gene is replaced by a mutant version of the same gene using homologous recombination. So far we discussed about deleting, but in the case of a knock-in, what you do, you actually instead of deleting the gene, you introduce a mutant version of a gene. This is very useful because if you want to actually study a disease model, for example, if you want to identify how a particular amino acid mutation of a particular protein is affecting the function of the protein. So, instead of deleting region, you can actually introduce between the two LOX sites a mutant form of the protein. So, on recombination, you will now generate a mutant, the wild type will be replaced by the mutant copy. Now, you can ask the question, what happens when the mutant protein is expressed? Will it produce the same kind of a disease as appears in the disease model? So, knock-in is also very useful in understanding the function of certain proteins that lead to certain diseases. So, more than 10,000 different genes in mice and approximately half of the genes in mammalian genome have been studied by using this kind of a knockout technology, and there is actually a roadmap to disrupt almost 20,000 genes in the mouse genome before the end of this year. There is what is called as a knockout mouse project, KOMP, which is actually funded by the National Institute of Health in the United States. Almost $4.8 million has been provided in June 2007, and if you can now go to the website www.komp.org, you can get what all the progress that is being made in this knockout mouse genome project. Here is another example, the European Conditional Mouse Mutagenesis Program. They have a year-wise target of how many genes has to be deleted in the mouse embryonic cells in a conditional manner or in a mixed manner, and year-wise they are given statistics on how they are going to go and disrupt genes one by one in the entire mouse genome. There are now a number of companies which can generate transgenic or knockout mouse. You just pay them, they develop the transgenic mouse or knockout mouse for you. Polygene, Ozgene, Vega Biolab and so on and so forth. And more recently, just this year, September 2010, the rat, knockout so far has been available only for mice, but now a knockout has also been developed using the homologous recombination technology. For example, the first paper appeared in this year, production of a p53 knockout rats by homologous recombination in embryonic stem cells. There are many other papers of similar nature. Why rat? Just like we have a knockout mouse consortium, now we have a knockout rat consortium, because many people think that rat is a better model, especially for certain physiologies and pathologies. For certain diseases, rat is a better model than a mouse. For example, rats have a heart rate similar to that of humans, whereas mice have a heart rate 5 to 10 times faster. Similarly, rats have been used as important model for human cardiovascular diseases, diabetes, arthritis. So if you want to study some of the genes involved in diseases, you would prefer to knock out those genes in rat rather than in mouse. So new technologies are being developed to knock out genes in rats. There is one other exciting development that has taken place, what are called as zinc finger nucleases that have been developed. As we studied in our earlier lectures, zinc finger transcription factors are sequence specific transcription factors. They have zinc fingers which go and specifically bind to target sequences and activate transcription. Example is steroid hormone receptors. So what they have done very cleverly, they take the DNA binding domain, which consists of 2 zinc fingers, and fuse it to the catalytic domain of a restriction enzyme called a FOC1 restriction nuclease. So this fusion between a sequence specific transcription factor, sequence specific DNA binding domain of a transcription factor and the catalytic domain of restriction enzyme creates what is called a site specific nuclease. This is called zinc finger nucleases. When you express these nucleases, this nuclease goes and specifically cuts near the target sequence. So it can specifically introduce double strand breaks depending upon the sequence which the zinc finger recognizes, and once you introduce a double stranded break by what is called a non-homologous end joining, these ends are repaired, and as a result you introduce extra bases and you lead to shift in the open reading frame and you disrupt the function of the gene. This has again has revolutionized. There is actually a company called Sangamo Biosciences. You can go to the company website and get more on how these zinc finger nucleases are revolutionizing the knockout technology. There are a number of papers on zinc finger nucleases. Due to paucity of time, I am not going to discuss. One can go through a number of review articles, a number of research articles that develop how these zinc finger nucleases are being used for knocking out genes right from plants, animals, across the animal and plant kingdom. I am also listing a number of key publications, especially from the Evans, Kaufman and Kepici's lab. Very important papers that actually revolutionized and that actually led to the award of the Nobel Prize for these researchers. Some of the review articles are listed here, and I am also listing here the Nobel lectures of Mario Kepici. You can just cut and paste this website. The entire PDF article gives you how he went on to develop this kind of a knockout technology. The Nobel lecture of Martin Evans, you can just click on this website and you can get the article, and again Nobel lecture of Oliver Smith is very useful how they went about and developed this technology. Also very important websites which gives you a lot more information about gene targeting and gene knockouts. I think I will stop here and hope I have convinced you that gene knockout has revolutionized the way research is done in biology and medicine, has given wonderful information about the function of various genes and has tremendous implications in the biomedical research. Thank you.